I am delighted to be able to share the work of our current postdoctoral fellows. We have one who was unable to present, but we have 7 presenting today. And as many of you probably know, in my role as the Assistant Dean for Research Operations and Training, I work pretty closely with our postdocs. And now I get to share with you how wonderful they are and all of the great work that everyone's doing. So what I'm going to do is first do a brief introduction of our postdocs, sort of who they're working with, who their mentor is, and then I'll let them talk about their research. So I will start with our brand spanking new postdoc, Yufeng Lin. She's been here a couple of weeks, maybe, Max. She works with Senhua Xiao. For those of you who don't know Senhua, she was assistant professor with us for a number of years, came in as a postdoc too under Deb Bruner, and moved to Yale, and now she's back. And so we're delighted to have her back and delighted to have Yufeng as well. So Yufeng, take it away.
Thank you. Let me share my slides. Can you see that? Yeah.
Yeah.
Yes.
Yeah.
Okay. Yeah. Sorry. Okay. Thank you all for being here today. Good afternoon, everyone. So my name is Yufeng Lin. I'm a new postdoc here, and it's my great pleasure to be the first to present today. So I will be presenting some of my dissertation research and some of my postdoc plans. First of all, I will briefly talk about my educational and professional background. I obtained my bachelor and master degree both in China, and I have 1-year internship at Xiangya Hospital and 2 years work experience as an oncology nurse at Shanghai Cancer Center. And during that time, I contact with numerous patients who diagnosed with GI cancer, and I saw them experiencing numerous distressing symptoms, which drove me my research focusing on this field. And I came to do for PhD degree, and I just graduated last month. And also during my PhD, I have several semesters of clinical shadow at Duke Cancer Institute and conducted my qualitative study and collect some data in the clinics. So my program of research mainly focus on 2 fields, symptom science and oncology nursing. So I use the NIH symptom science model to guide my research trajectories. During my pre-doc training, I mainly focused on the first 2 goals to describe and identify complex symptoms, and also using the demographic and clinical characteristics to characterize different phenotypes among these symptoms and patients. And then for my postdoc training, I will move forward to the next 2 goals, mainly focused on the discovered biomarker and using the omic science to understand the biologic mechanism underlying these multiple symptoms, and then to provide some directions for the like intervention, development, and clinical applications. So my dissertation title is Multiple Co-occurring Symptoms in Patients with GI Cancer. I use the multiple co-occurring symptom model to guide my dissertation research. For my chapter 2, I use integrative literature review to cover all 6 symptom-related concepts in patients with gastric cancer. And in chapter to create a deeper understanding of the symptoms in these patients. And in chapter 3, I conducted a literature review, qualitative study to describe the symptom experiences such as symptom occurrence, severity, distress, frequency, and meaning, and their symptom self-management strategies for multiple co-occurring symptoms in patients with gastric cancer. And in chapter 4, I used a person-centered approach to identify the distinct symptom subgroup of patients with distinct symptom profiles of multiple co-occurring symptoms, and then to evaluate the predictors and outcomes among these different subgroups. And in chapter 5 and 6, I mainly focus on the trajectory of the morning and evening fatigue and sleep disturbance. Also use a similar a person-centered approach to identify the subset of patients with distinct symptom trajectories and also over the 2 cycles of chemotherapy, and to evaluate the predictors, the other co-occurring symptoms such as pain, anxiety, depression, cognitive dysfunction, and symptom outcomes among these subgroups. And to date, all those 5 chapters were published, so you can review them online for further information. And among these 3 chapters, I use the same data set that is a project collaborated with UCSF. It is part of a longitudinal study, and we collect 6 time points data over 2 cycles of chemotherapy. I use the subset of patients with GI cancer. And they were more than 18 years old and had received chemotherapy within the preceding 4 weeks and scheduled to receive at least 2 cycles of chemotherapy. The sample size were 405, and patients complete service at home and return them in a posted paid envelope. We use the latent class or latent profile analysis to identify the subgroup of patients with distinct symptom profiles, and use the parametric and non-parametric tags such as ANOVA, Corvallis to evaluate for differences among subgroups. And currently, I'm still working on this data set to work on another 2 or 3 papers using this data set. And then I will briefly talk a little bit about my postdoc plans, because I just started this month. So I cannot make a timeline for my 2 years postdoc training. And first, I will focus on the grant writing. I'm going to submit the K99RO grant for the February 2022 cycles. In the meanwhile, I will consider to submit other small grants such as Hillbrow Nurse Scholar Award, Oncology Nursing Foundation Research Grant, and other internal external grants opportunities, such as Winship Cancer Center, some small grant, and also focusing on several manuscript development. Currently, I'm working on 2 first author paper. One is using Dr. Xiao's dataset to focusing on the psychoneurological symptom clusters and the gene impression about the association between them. And also, like I said before, I also working on my dissertation data set to further identify the distinct profiles of co-occurring symptoms, co-occurring morning and evening fatigue and other symptoms in patients with GI cancer. And also, I have working on several co-author papers. That is primarily my dissertation project. I was involved in an educational project on simulations, and also 2 other projects about the expressive writing in cancer survivors and breast cancer symptom management, and also about the social network interventions. And in addition, for the research dissemination, I would like to attend several international and national and regional conferences such as MASC. It's a multinational association of the supportive care in cancer, and also other like genetic association conference, etc. And also like Oncology Nursing Society and other national conferences. Probably I planning like 2 conference each year. And then I will attend several trainings and workshops to further acquire knowledge and skills in this field. I have signed up the Washington University Summer Institute in Statistical Genetics to further learn about the genetics and genomics analytics. And also considering to auditing the Omics in Health and Disease course for the fall semester, and other opportunities such as microbiome bootcamp and like grant writing workshop had by the OP, this kind of trainings. and others, including the teaching and service and clinical plans. I'm not sure if I have time for that. And currently I was involved in a teaching project that is an international collaboration project. So it's mainly focused on transcultural nursing through a virtual simulation. And we were going to have the summer course this year. And other teaching like oncology or research methodologies courses, I would like to co-teach or being the teaching assistant. In the meanwhile, I was on the 3 journals peer reviews and would like to attend other service. Yeah, I think that's it. Thank you all for listening. I'm very happy to take any questions.
Thank you, Yufan. Sounds like you've got a busy 2 years ahead of you. Nicely done. What questions do folks have for Yufan? Words of advice or questions, anything like that? Okay. We will move, thank you, Yufan. We will move on to our next presenter. We have Madeline Hauser, who has been working with Vicki Hertzberg. Now, Madeline is near the end of her postdoctoral fellowship. And so we will let Madeline take it away and tell us about what you're been doing and what your future plans.
All right. Let me get this going. All right, can everyone see that okay?
Sure can.
Great. Well, good afternoon. Thank you all so much for coming. My name is Madeline Hauser, and I'm working with Dr. Vicki Hertzberg. And I hope you all will excuse my gravelly voice today. I'm getting over a cold and trying not to lose my voice, so I hope you'll bear with me for the next few minutes. So to give you all, first of all, for those who may not know me, a little bit of my background or a reminder for those who do. Before starting my postdoc in the School of Nursing, I did my PhD at Emory as well, over in the School of Medicine in the Immunology and Molecular Pathogenesis program. And the focus of my research dealt a lot with the gut-brain axis, and my dissertation work was specifically on the contribution of gastrointestinal inflammation to the development of Parkinson's disease. But during that time, I was also involved in a lot of different collaborative projects. And in the course of my graduate time, I was an author on 5 published research articles, one of which was first author, and 4 reviews, one of which was first author. After I defended, I helped my graduate lab transition to a new institution. I did a brief postdoc there and then started my postdoc here in the School of Nursing in November 2019. And since then, I have been involved with, or I'm still actively contributing to, I think about 9 different projects on various different topics, many of which did tap into my knowledge of the immune system and inflammation, neurological disorders. But I've also had an opportunity to really dedicate myself to improving my understanding of different statistical methods and methods for big data analysis, particularly microbiome and metabolomics. And since I started my postdoc here, I have been an author on 4 published research articles, one of which is first author, 2 more currently under review, as well as 1 review with 1 more submitted. And as I said, I have been involved on a lot of different projects. So I wanted to take this research in progress talk opportunity today to tell you about just one of those projects and some of the interesting findings that came out of it. And this was published just last month in Biological Research for Nursing. So to kind of introduce this, as I'm sure all of you know, it gets quite hot here in the Southeast in the summer. And I imagine that most of us are very grateful that we can do our work primarily from air-conditioned spaces, but that's not an option for everyone, including agricultural workers here in the Southeast. These workers are exposed to frequent, intense heat exposure during their work shifts and exertion in the heat. And in addition to increasing their risk for heat-related illnesses, the kind of the classic heat-related illnesses that we recognize, there's also concern that this may increase their risk for acute kidney injury and ultimately for the development of chronic kidney disease. And the mechanisms that are driving this kidney injury are not really well understood, but it is thought that dehydration maybe plays a role, and there is evidence to suggest that an inflammatory mechanism might be involved. So the idea behind this study was to use both direct comparisons with kind of classical statistical methods as well as machine learning methods to try to identify potential biomarkers that are associated with dehydration and acute kidney injury in a cohort of Hispanic agricultural workers in Homestead, Florida. So the design of this study was that at what we called the baseline, so on a non-work day, we measured indicators of dehydration, inflammation, muscle damage, and kidney function in the cohort of workers. And then over the course of their next 3 days of work, the heat index, the workers' core body temperature, and their heart rate were all measured. And before and after their work shifts, measured indicators of dehydration and kidney function. And I'll just say thank you to all the co-authors on here who were actually the ones conducting this part of the study before I came in later and took over the data analysis portion of it. So some of the interesting findings that came out of this were, first of all, that when we looked at the participants who were dehydrated at baseline, so on a non-work day, we found that over 2/3 of the participants were dehydrated, which is itself concerning. And the factors that were most associated with dehydration at baseline included a history of hypertension, history of diabetes or abnormal glucose readings, as well as low levels of uromodulin and sodium in urine, and high levels of IL-6 and C-reactive protein, or CRP, in serum. And it's not terribly surprising that we would see associations with urine analytes in dehydrated participants, but this low uromodulin is actually quite interesting because uromodulin has anti-inflammatory properties, and high levels of uromodulin have actually been shown to be protective against kidney injury. And we're also seeing this inflammatory profile in dehydrated subjects, which could relate to the history of some of these comorbidities that we've seen associated with dehydration, and could indicate a inflammatory mechanism that's linked to dehydration that could impact kidney injury. And so when we look at the participants who did meet the criteria for acute kidney injury after at least one of their work shifts, this was about half of the participants in the study. We saw that the factors that were most predictive of developing kidney injury were having a high BMI, being dehydrated at baseline, having low levels of uromodulin in urine at baseline, and high levels of IL-6, CRP, and lipopolysaccharide binding protein, or LBP, which was correlated with these other inflammatory factors, in the serum at baseline. So we can see a lot of overlap between our dehydrated subjects at baseline and our subjects who went on to develop AKI. So this definitely suggests that dehydration is potentially playing a role in this kidney injury, and that there may be this inflammatory mechanism involving IL-6 and CRP, as well as LBP, which points to some gut leakiness potentially involved. And so this may be an inflammatory mechanism that's linking dehydration and acute kidney injury in agricultural workers. So just a couple of notes on some ongoing research projects. The next step in the cohort of agricultural workers that I just told you about is taking some untargeted metabolomics data that we already have collected and analyzing that, looking for associations between particular metabolomic profiles and dehydration and kidney injury in these workers. And then I'm also managing a longitudinal study in which we're collecting samples from ALS patients and their spouses who are serving as controls, And we're handling this study essentially all remotely since COVID started. And subjects are sending in stool and saliva samples for microbiome analysis and metabolomics. And we've got about 15 couples that have been consented and are actively participating in that study. And then I've also joined the data science core team for the P30 Center for the Study of Symptom Science, Metabolomics, and Multiple Chronic Conditions. And in this role, I'm responsible for processing and analyzing the untargeted metabolomics data that is associated with the various different projects under the P30 umbrella, and also working with the microbiome data for that. So I am keeping busy, and I would be happy to answer any questions if you all have them.
Great, thank you, Madeline. What questions do you have for Madeline? Yes.
Hi, Madeline. This is Talisa Spice. Good talk. I just have a question. How did you go about choosing your inflammatory biomarkers? You know, because a lot of them are non-specific. So I was just curious as to how did you decide which inflammatory biomarkers you all wanted to observe?
Definitely. So we looked at essentially a panel of common inflammatory markers and measured all of them using multiplex immunoassay. So we looked at some of the kind of common suspects that you see, like CRP, as you say, is kind of a non-specific indicator of inflammation. But we were actually particularly interested in this kind of IL-6, CRP, LBP pathway because there have been studies that were reporting that under conditions of heat stress and physical exertion under heat stress, that this can induce gut leakiness, which can promote systemic inflammation. And that had actually been kind of proposed as one potential mechanism that may be involved in heat-related illness and potentially acute kidney injury. So that was actually something that we were interested in looking at, but we did look at various other markers, CXCL8, IL-10, IL-1 beta, that sort of thing.
Okay, thank you.
Thank you. Okay, I wanted to announce that we had a bit of a scheduling snafu, and in fact, we have 8 folks presenting today. So now we need to squeeze in one more person. So I'm going to go ahead and move on. Thank you very much, Madeline. We're going to go ahead and move on to Vivian. Vivian is also nearing the end of her postdoctoral fellowship, and she has also been working with Dr. Vicki Hertzberg, although in a different way. So we look forward to hearing about your work, Vivian. Vivian, you're on mute, dear.
Sorry. Thank you for attending my research in progress roundtable presentation today. My name is Wenhui Zhang. People call me Vivian. I'm currently a postdoc fellow and will be a research assistant professor at Emory School of Nursing. My mentor are Dr. Vicki Hertzberg and Dr. Roy Simpson. So I'm going to present my research focusing on optimizing health and life of people aging with multiple conditions. And my goal is to contribute to more strategic actions in the care for these patients based on my aging, nursing, and statistic training in the past. Yeah, so next slide. Yeah, so here is the overview of the presentation today. I'm going to briefly talk about my research topic, background, research questions and approach in the past and up to date, my accomplishments in the past year, and my future research, hopefully to leave for 3 minutes for questions and answers. So talking about multiple chronic conditions, it is well known that multiple chronic condition is related to age and contribute to high burden to the patient, family, and healthcare system. So based on the Health and Human Service Department's strategic framework, I have been learning and studying mainly on self-management in my PhD work. And then because of my statistic training, I was also exposed to research using electronic health record and population level data that found that multimorbidity contributed to patient outcomes and healthcare quality. So nowadays, during my postdoc, I start to look at whether I can apply electronic health record and machine learning to develop tools to inform and improve the care for individuals with multiple chronic conditions. I also start to look at biomarkers and omics research that could fill in some knowledge gap for these patients. So talking about multiple chronic conditions, the American Geriatric Society developed this paper from a panel of experts, and they highlighted that as the number of chronic disabling conditions increase, there are more uncertainties in the care for these patients. So there's a need for more research and explorations there. So following that, my PhD dissertation aimed to study the self-regulation strategies of selection, optimization, and compensation in the self-measurement of individuals with arthritis and multimorbidities, and found that the health resources, especially the communication quality with healthcare providers and the chronic disabling symptoms, especially the physical disability symptoms, contributed together with the self-regulation strategies to the self-efficacy and then the quality of life. It also identified like gaps such as like that were not well studied in my dissertation, such as the interaction between the chronic conditions and also how this like omics could contribute to the disabled symptoms. Mm-hmm. So following that limitations and gaps of knowledge, I'm now conducting a study focusing on African Americans with arthritis and hypertension, because hypertension was not well measured by the functional comorbidity index measure in my dissertation. So I'm now recruiting participants. I'm aiming for 80 participants and have collected data from 20 already. And then the other research question raised based on my analysis with electronic health record and my work with Dr. Herzberg with the EHRs in Emory and MIMIC3, I developed a K9 ROO proposal aims to study delirium, because delirium is one of the key nursing quality indicator, and also people with multimorbidity and dementia are at higher risk of having delirium during hospitalization. So the goal of this K9 is to use electronic health record and machine learning to predict delirium risk, detect delirium early signs and symptoms, and predict delirium outcomes downside the healthcare trajectory. And then the goal for the future is to develop these clinical decision support tools to inform the prevention, screening, and care for people at risk of delirium or have had delirium. So to achieve my current research goals and future research goals, I obtained more training in such as in microbiome analysis, teaching, aging and multimorbidities, smart and connected health, delirium, and aging, mental health, and biostatistics in the past year. I've also followed my PhD research and postdoc research roles and developed a few publications. Some of them are in revision and in review. I've also I was also very fortunate to be funded together with Dr. Bai and Dr. Kim and my mentors studying the mental health among Chinese and Korean immigrants, and start to learn how to launch a new course on cultural immigration and health. So my future research will be based on will follow my previous research interests and based on some of the proposals I've written in the past year, such as focusing on the metabolic and chronic disability symptoms among African Americans with rheumatoid arthritis and hypertension, gut microbiome and delirium, multimorbidity and healthcare trajectory phenotypes, and delirium in COVID. Please let me know if you have any questions. Thank you.
Thank you, Vivian. Great work. Wonderful presentation. Thank you. Okay, who has questions and what are they? This is like speed dating, right? It's a lot of information. You guys have been great. Very, very busy. Any questions? Okay. Thank you, Vivian. And next up, we have Zara, and I want to butcher your last name, so please correct it. It's Barandosi. And you work with Dr. Deb Bruner. And Zara, you are in your, let's see, what day and month is it? You're still at the end of your first year. Yes. Okay. I have to think for a minute. Okay, go ahead and tell us about your work, Zara. Thank you.
Sure. Hello, everyone. Thank you for being here and listen to my presentation. So I'm postdoctoral fellow here. So for this presentation, I'm going to talk a little bit about my background, my research trajectory, and my current and future research plans. So I studied midwifery in Iran and then come to the US to get my PhD in nursing at University of Connecticut. And last year I graduated and started my postdoc position in last year, August here. So in terms of the research trajectory, so during my PhD program, I focus on understanding the biological mechanism of the symptoms in people with irritable bowel syndrome. So for doing that, specifically, I look at the association of the gut microbiome and neurotransmitters with anxiety and depressive symptoms in this population. But after coming to the Emory and started my postdoc, so my population has changed to cancer population. So I realized I need to pay attention to the trajectory of the symptoms in a cancer patient and specifically gynecologic cancer patient because my mentor most specifically has project in her hand actually, and specifically studies on the gynecologic cancer patient. Based on my literature search and understanding, I realized that and based on the report of the American Cancer Society, advances in cancer screening and early stage diagnosis has improved overall survival rate in gynecologic cancer patients. However, this population experienced various symptoms such as pain, fatigue, sleep disturbance long after treatment. So these symptoms can cause by cancer itself or can cause by different type of treatments such as chemotherapy, radiation therapy, or chemoradiation. But these symptoms can negatively impact patients' quality of life, functional status, and even survival rate. So at this moment, I realized, okay, this is a trajectory of the symptoms in cancer patients. So what is the next step? So do I need to study the just single symptoms or cluster of the symptoms? So again, I look at the literature and I realize that the researchers have both directions. Some of them just look at the individual symptom, focus on the understanding the biological mechanism of just single symptom, and some of the researchers just focus on the cluster of the symptoms. And by exploring the data set that I had in my hand, for example, I look at the depressive symptoms and predictors in women with gynecologic cancer, and at the same time, I look at the to understand the longitudinal change in the psychoneurological symptom cluster in gynecologic cancer patient. I realized that it is better for my research to focus on the cluster of the symptom rather than focusing on individual symptoms. In terms of the mechanism, so I try to, because from my PhD program, I focus on the biological mechanism of the symptoms. So for my postdoc, I try to relate the gut-brain axis as one of the contributing factors underlying symptoms. So emerging evidence shows that the cancer treatment or even cancer can change the gut microbiome, and changing gut microbiome profile can disturb the bidirectional interaction between the gut and brain. And the interesting finding that I realized during my literature is that the immune system is one of the systems in the gut-brain axis signaling that may communicate between the brain and gut. So focusing on the gut microbiome and immune system as the two important part in the gut-brain axis may clear our understanding about the mechanism underlying symptoms. So for doing that, I try to actually use the database in our hands. So I look at the association of the depressive symptom with gut microbiome in patient with head and neck cancer by using the Dr. Zhou data set. At the same time, I try to look at the association of the psychoneurological symptom cluster with inflammation in this cancer population. For moving forward, because my cancer population will be gynecologic cancer patient, I try to look at the association of the psychoneurological symptom with gut microbiome and inflammation in this gynecologic cancer patient. So I submitted grant and as well as look at the literature to make a kind of the foundation for my idea about the association of these two important parts of the gut-brain axis in development of the symptoms. So at this moment, based on my research hypothesis or ideas, so I submitted a grant for the Oncology Nursing Science Foundation grant, Sigma Data, and I'm going to submit for K99. At the same time, I'm trying to go more in depth and realize the symptoms that symptoms that co-occur in gynecologic cancer patient by doing the factor analysis, and at the same time try to understand the association of the immune system and the gut microbiome in development of the cluster of the symptoms. Also, for doing that, I prepared some manuscript by using the Dr. Sanwa data set, as well as a systematic review that I actually did with the collaboration with my team, our team here. And also I presented some of this research in some of the conferences, Oncology Nursing Society, MAS, and World Microbiome Forum. So this is my research trajectory at this moment, and I'm trying to more focus on this association by working on the available data set and publishing more manuscripts. Thank you so much for your time.
Thank you, Zara. Excellent. Excellent. Any questions for Zara and the work that she's been doing? This is a very, very busy group. Very productive.
Hi, Zara, I had a question if you have a sec.
Sure.
So really, really interesting work and interesting proposals. I'm curious on your connections between cancer and microbiome and symptoms. Are you thinking that, are you kind of looking at it as essentially cancer is impacting the microbiome and microbiome is contributing to these symptoms, or are you thinking that, you know, particular individuals with a particular microbiome going into a cancer diagnosis or something may be more or less likely to develop particular symptoms, or kind of what framework are you taking with it?
Okay, it's a good question. So based on the literature, cancer and specifically the cancer treatment can actually disturb the gut microbiome profile. And at this moment, the knowledge that we have is that when the cancer patient receive the treatment, the gut microbiome profile is different from the healthy people. So what I'm working here, so my target is to look at the association of the gut microbiome in people that receive specifically radiation therapy, in cancer patients that receive radiation therapy, and see what is the change happening in their gut microbiome profile. And at the same time, I'm going to have the healthy people as a kind of the standard to see, okay, if this is a cancer patient, in which time point their gut microbiome profile change compared to the healthy people. I think by looking at this, at the research at this viewpoint, we can actually be clear about what's going on there. Now, did I answer your question?
Yeah, that's great. Thanks. Best of luck.
Thank you.
There are no more questions, and we will move on to Kalisha Bonds. And Kalisha is a postdoctoral
Alicia
fellow.
Johnson.
Liz Bonds Johnson, I'm sorry.
It's fine. It's taken a while to
I've
take.
known you as Kalisha Bonds for far too long, so I get set in my ways. Kalisha Johnson, Kalisha Bonds Johnson, has been with us for 2 years, and she is one of our T32 postdoctoral fellows, and her mentor is Dr. Ken Hepburn. So we look forward to hearing about your work, Kalisha.
Thank you. And I too am learning that it's Bonds Johnson or Johnson, so it's fine. I'm just giving you a hard time because I love you, Jenna.
I know.
So thank you, everyone, for being here. My talk is about improving the quality of life of African-American dementia dyads and families through decision-making, really focusing on two sort of lines of inquiry, those around healthcare providers and those around adult donors. So of course, the significance of my work, dementia is a multifaceted diagnosis. It causes cognitive impairment, it affects social functioning, physical functioning, one's ability to care for themselves. And while the exact number of persons living with Alzheimer's disease and related dementias is not known, we do estimate that it's around 6 million people that have this diagnosis. Dementia has been more recently considered the third leading cause of death in older adults. And there is an increased risk for African-American older adults. It's suggested that African-Americans are 2 times more likely to develop Alzheimer's disease and related dementias when compared to non-Hispanic white older adults. As we know, there is no cure for dementia or Alzheimer's disease, so the sort of gold standard is to treat with medication earlier in the disease process so to help slow the progression of these symptoms. Yes. Given how African Americans entered the United States with the mistreatment that has occurred both in medicine and research, many African Americans are often hesitant to seek healthcare services and thus are diagnosed later in the disease trajectory. The Alzheimer's Association did their typical yearly facts and figures, and this year focused a little bit more on discrimination and racism. And 50% of Black Americans reported discrimination as a barrier to Alzheimer's disease dementia care. And only 53% of Black Americans trust that a future cure for Alzheimer's disease will be shared equally, regardless of race, color, or ethnicity, highlighting a need for research in this area. Specifically in my own work, I have included the African-American older adult, recognizing that they often have a unique role in families, and that when we think about cultural norms of African-Americans, older adults are often highly respected. So I shared this in my job talk, but it's just sort of my career trajectory regarding grants and awards and things of that nature. This trainee award I did receive, and we'll go into greater detail about that in the next couple of slides. The NIH and the AK23, I'm still waiting to hear, but hopeful as my just-in-time materials were requested and they are due tomorrow. So I'm hurriedly working to get things submitted to IRB as I need to show that that has been done by tomorrow. This NIH Impact Collaboratory, again, these are just future grants, and I'll go through it a little quickly, just sort of show my trajectory of where I think my research will go in different funding mechanisms I intend to use. So the ADRC REC, or Research Education Component grant proposal, was recently awarded to me. I have come up with sort of a clever name, I think, when it comes to my lab or future lab and studies, and that is, I really wanted to do a play on decide, since I'm really interested in how decisions are made and the decision-making processes. So decision-making experiences for culturally inclusive dementia engagement, or decide, is what I'll call my lab. And then this REC grant will focus on providers. So it's decide providers, formal care or healthcare services from a paid institution. Another way to describe formal care is essential to the treatment and management of persons living with Alzheimer's disease and related dementias. However, African-American persons living with dementia often have low rates of formal care use, health disparities in receiving quality formal care, and under treatment of their dementia symptoms. which result in great morbidity and worse quality of life. While we know little about the Alzheimer's disease, while we know little about the dementia dyad, we know even less when it comes to healthcare perspectives and providers' input. So the purpose of this grant is it's a qualitative descriptive study to explore the firsthand accounts of formal care providers, and I define those as physicians, nurse practitioners, physician's assistants, and staff, so nurses, social workers, and medical assistants, their experiences in engaging with formal care decision-making for African-American or Black persons living with Alzheimer's disease and related dementias, and their caregivers and families. The K-23 grant proposal that I received an impact score of 20 on from NIA is a 5-year project, and we'll focus on the perspectives of the dyads and families. In my dissertation work, I discovered that it seems a subgroup of adult daughters who are the primary caregivers of African-American persons living with dementia. They, as well as the parent they provide care for, experience worse quality of life when compared to African-American couples. And so I focus more specifically on this subgroup and how they navigate formal care services. The purpose of this convergent mixed method study is to design and test the feasibility of a culturally tailored prototype intervention to support the formal care decision-making process for African-American persons living with dementia that positively influences the quality of life of African-American parent, adult, daughter, dementia. So that's pretty much what I'm working on now. I will say that ADRD grant has been submitted to IRB. This will be submitted to IRB today, to the list. But thank you, and if you have any questions.
Thank you, Kalisha. Wonderful work. What questions do we have for Kalisha?
Hi, Dr. Johnson. This is Dion Williams. I'm a
Dion, you can call me Kalisha.
Hi, Kalisha. How are you? I do have a question. Among your population, have you considered cultural factors such as spirituality or religiosity among the decision making among African Americans? And also, have you considered those cultural aspects having some type of role in caregiving?
You know, I looked at that a little bit in my dissertation. This is why I need you to finish your PhD so that we can collab, because that is your area of expertise. But no, I think you bring up a good point. And I think spirituality and religiosity have, you know, there's key cornerstones of African-American culture. And while I haven't specifically focused on it, I think it would be a great addition to my work. And like I said, I need you to finish that case so that you can, so that we can collab. Thank you.
Soon come. Thank you.
Great question. Thank you. Other questions for Kalisha? Dr. Johnson.
Bonds Johnson.
Bonds.
I'm not
Are you
hyphenating?
Okay.
Away.
Well, I hear you. All right. We'll talk later. Okay. Thanks. Okay. Next, we have another one of our T32 postdoctoral fellows, Catherine Breen, and she is wrapping up her first year in the T32. Her mentor is Dr. Becky Gary. And Catherine, we look forward to hearing about your work.
All right, thank you. I'm currently locked out of the OneDrive. I've been on the phone with IT to try to get slides for Emory, so that's why this is the way it looks. So I apologize for that. Yes, I'm wrapping up my first year, and I'm just going to go over a few of the things that I've been up to, future directions, and where I'm at. So my research updates, just some basic updates. I have currently submitted abstracts to both AHA and SNRS for the 2021 conferences. I will find out if I was accepted to the University of Utah's grant writing study around July 5th, sorry. Currently working on 3 manuscripts, and I'm attending a Writing for Scientific Impact. And I will see you there at the Summer Institute in Statistical Genetics. I will also be taking 736 quantitative analysis in the fall. I have just recently collaborated with Dr. Hertzberg, and I'm going to join the Data Science Corps as well. And we will be working together on metabolomic papers in the fall. And I will be moving to Atlanta at the end of August for at least the fall semester. to try to facilitate some of the difficulties I've had trying to get my research up and running being remote. My future directions, I'm currently going back and doing a new analysis on my postdoc or on my predoctoral work, which was in multimorbidity phenotypes that were predictive of acute coronary syndrome. And I'm going to examine differences in symptom presentation by multimorbidity phenotypes. My new directions that are not part of my predoctoral work is I will be using machine learning to conduct a biomarker-based clustering analysis to identify subgroups of cardiometabolic multimorbidity in older people living with HIV using Dr. Gary's Fit Brain study and Drena's as well, sorry. And I will also be doing that in coronary artery disease. And I will, with the end goal, I want to change the CVD risk stratification scores that we have. They were never originally designed to be used in people over the age of 65. And to tell you the truth, from what I've been reading in the literature, they do not perform very well. My first study, I submitted this for Healrun, which was unfortunately not funded, and I'm getting ready to resubmit it to SNRS for their grant, is the effects of regular pro-inflammatory plasma microRNA profiles in obese people living with HIV. My samples are currently at Yerkes and being analyzed right now. It has been a journey, but I'm happy to be here. And my overall— Yay! Now, Drenna and Becky were both in on that with me. So I'm happy to report that that's good. So we're getting those results back soon and writing them up, and hopefully we'll get at least 2 manuscripts out of it by the end of the fall. My overarching thought is that transcriptomic profiles impact cognitive function, and that is through their control and regulation of pro-inflammatory cytokines, which increase inflammation and all of those other things in the figure, and that exercise will decrease inflammation, and that the benefits of exercise are contained within the exosomal microRNA. And these are my aims of that study, and those, I won't bore you with all of the hypotheses and things like that, but I just posted it. I wasn't really sure about what was supposed to be on this presentation. I thought it was pretty casual, but I apologize for that. So that's kind of what I'm doing. And I'm also going to be rekeying this to look at metabolomic profiles. I'm currently getting pricing and structure on that to identify subgroups of these patients and their divergent outcomes. This is my abstract I have submitted to both AHA, and I submitted a different version of it for SNRS. I was able to, through my big data course, use machine learning, a logistic regression model to identify in a subset of 414 people that TNF alpha is associated with being multimorbid. My discrimination was okay, but it is a positive sign that biomarkers are based on multimorbidity phenotype. And I'm going to be using multimorbid machine learning to identify multimorbidity phenotypes predictive acute coronary syndrome. I'm currently in the data analysis phase of this, and I'm redoing my predoctoral study to validate my phenotypes that I found, which just to do a little bit of review, I found 4 phenotypes. There was a high overall one, a low overall multimorbidity, a cardiovascular, and a cardio-oncology group. The cardiovascular and the cardio-oncology group were more predictive of being ruled in for acute coronary syndrome. Currently, one of my methodologies, the KNN, in the very preliminary stage, has shown that there may be more of an underlying autoimmune factor to the cardio-oncology group than my latent class analysis was previously able to identify. So that's interesting. And hopefully more on that to come soon. And then my K23, there was a bit of a hiccup, and I just got relinked up with the Emory Cardiovascular Biobank. My tentative submit date is October 2021, and the title is Using Machine Learning to Identify Metabolic Subgroups of Multimorbidity in Older Adults with Coronary Artery Disease. My mentorship team is, as it stands now, is Rebecca Gary, Vicki Hertzberg in our shed, and Joyce Ho is my computer scientist, and Melinda Higgins is my consultant. And then my aims are to identify multimorbidity profiles in patients with CAD versus patients without CAD and changes in these profiles over time. I'm going to use a metabolomic-driven analysis to identify metabolites and metabolomic pathways associated with CAD in the context of these profiles. and those will be my subgroups. And my exploratory aim is to explore the relationship between multimorbidity profiles and clinical biomarkers at baseline. I currently have a meeting on Thursday with their bioinformaticist to actually get what is in the data set. So these aims are very preliminary. And other grant submissions, as I mentioned, my SNRS, I've got one to HA, and I will be resubmitting to Heilbronn to use machine learning to examine the impact of regular exercise on plasma metabolic profiles in patients with cardiometabolic multimorbidity. Thank you guys for your time.
Thanks, Catherine. Great work. I'm excited to hear that Yerkes has come through. Any questions for Catherine?
Hi, this is Sandy Dunbar. Quick question for Catherine. And that was a great presentation. I wonder, Catherine, I'm really glad you're looking at phenotypes, and we looked at this in relationship to looking at self-care behaviors and outcomes. And so, you know, the obvious answer is that the more comorbidities you have, the worse your self-care is going to be. But we found that it was certain patterns of comorbidities that affected self-care more. So I guess my question to you is, what do you think about your patient outcomes? You primarily have event outcomes with morbidity, mortality, and events. And I'm just wondering if you might look at other types of patient outcomes in terms of quality of life or other markers.
If I stay in the Emory Cardiovascular Biobank, which is my tentative plan now, the only thing is I have an angina, the Seattle Angina Questionnaire, and that only has one quality of life metric. They didn't really measure that. That wasn't their outcome goal. They were just looking at cardiovascular events over 1, 3, and like 5 years. But that is a good point. I would eventually like to get it to self-care behaviors and interventions. But given the data set and that I'm using, it's just not going to happen. If you've got data, I would love to analyze it.
Well, I don't think I have the biomarkers that you want, but certainly the other components.
Okay.
We can look at. Yeah. Thank you.
Thank you.
Thanks. Great question, Sandy. Other questions for Catherine? Okay. Thank you, Catherine. Okay, next we will move to Gia Daniels. Gia is working with Dr. Alicia Johnson as her mentor. Her T32 postdoctoral fellowship is through the School of Public Health. And so probably true to the way many of us work, she's got arms in a number of pots. And so really exciting work, and we're looking forward to hearing your updates, Gia. Thanks.
All righty. Hi, everyone. As Drina said, my name's Gia Daniels, and I am a postdoctoral fellow, second year. And so for those of you who don't already know me, just a little bit of background. For my dissertation work, I focused on how health behaviors like intravaginal practices and social determinants of health like education and insurance affect the vaginal microbiome. I also explored associations between the vaginal microbiome and vaginal symptoms. And learning about the vaginal microbiome and how the literature presents a difference in the microbial composition between Black and white women has magnified my curiosity in sexual health. So many of you have heard me say over and over again that race is social, not biological. And because of this, the color of one's skin does not predict the bacteria in one's vagina. Therefore, researchers should seek to explain differences in outcomes beyond race. And my research interests have evolved to focus on sexual health outcomes in Black women that present as race-based disparities. Let's see. So my current role, after completing my doctoral program, I obtained my current position as a postdoctoral fellow in the Environmental Health Science and Toxicology NIEHS T32 program at Rollins School of Public Health. And the structure of this program allows for me to stay in the School of Nursing since my advisor, Dr. Kalisha Johnson, is here. So I've always desired a two-pronged approach to my research, and that's health disparities and increasing diversity in nursing. My dissertation work and speaking with audience members after conference presentations inspired me to focus on designing a mixed method study that describes what social cultural factors influence sexual health behaviors of Black women, as well as examining the toxic effects that those sexual health behaviors have on their vaginal environment. In order to develop an appropriate and relevant intervention, I want to develop a thorough understanding of Black women's perceptions of their sexual health behaviors. And because I value interprofessional collaboration, I've established a network that extends beyond the School of Nursing to achieve my goals. In addition to Dr. Lisa Thompson in the School of Nursing, I've worked very closely with Drs. Anandi Sheth and Christina Mehta in the School of Medicine, and also Dana Boyd Barr in the School of Public Health, as well as others to prepare a grant application to ultimately integrate qualitative and quantitative findings to assess associations between vaginal hygiene and sexual practices and product use and environmental toxins in Black women. Additionally, I've been working with a colleague at Mercer University, Dr. LaDanya Patterson, to develop and implement a pipeline program for Black men to facilitate entry into the nursing profession called High School to Higher Education. Our program received the Nursing Innovations Fund Award through the Robert Wood Johnson Foundation, AARP, and AARP Foundation. And the School of Nursing serves as one of our community partners. We'll be conducting a mixed method study to evaluate how a program like ours can support increasing the number of Black men in nursing, and our IRB application was recently approved to do so. In the spirit of success in the academy, I am first author and co-author of accepted manuscripts and poster presentations. I've submitted a grant application for my work in sexual health disparities, which I'm still waiting to hear about. I've received funding for my work with increasing diversity in nursing, and I've interviewed for a faculty position here in the School of Nursing. As you can see, I've been very busy during my fellowship and look forward to transitioning into a faculty position here in the School of Nursing to continue my work in improving sexual health disparities and increasing diversity within the nursing workforce. Thank you.
Thank you. Great work, Gia. Great updates. Questions? I apologize. I use everybody's first name because we're like a family.
That's true. That's true.
Okay. Okay, if there are no questions, then we definitely have time for Despina Tesmenti. She has been with us, gosh, you're in your third year now, correct? With your postdoc, been working with Dr. Bruner, and we had a bit of a scheduling communication snafu, but all is well, and she's here with a presentation, and we look forward to hearing from you and getting an update on your work.
Thank you. Can you hear me well?
Yes.
Okay, great. Sounds great. Thank you very much for having me. And thanks everybody for the time attending our presentation today. Am I sharing?
Yes, it's not in like the presentation mode, but you're sharing.
Okay.
There you go.
Okay.
Yeah.
There we go. So my name is Despina Tesmenti. I am originally from Greece, where I studied biology, and I obtained my Bachelor of Science in Biology with a specialization in genetics, molecular biology, and biotechnology, where I used to work on genetic engineering in plants, so very far from nursing. But I switched gears completely when I decided to study microbes. So I came in Atlanta back in 2010, and I obtained a PhD in environmental engineering with a minor in microbial ecology, evolution, and ecogenomics from Georgia Tech, which is essentially the study of microbes in their natural environment, or what we collectively call as the field of microbial ecology. And as all microbial ecologists like to say, we live in a microbial world, and microbes affect processes ranging from our health to global biochemical cycles. And this is why we want to understand how microbes live together and form communities, and the role that they have, for example, in their human host, with the ultimate goal to predict or even manipulate their functions. And how do we do that? We are lucky to live in the sequence era, where we don't have to isolate and culture microbes in the lab one by one, but we can instead use what we collectively call omic technologies, or the ecogenomic toolbox, which means that we go to any environment, including clinical settings and the human body, collect a sample that contains the microbe, sequence the total DNA, and use bioinformatic tools to figure out who is there and also their functional potential, what they're doing at the different levels, either at single organisms, population, or community. And using this ecogenomic toolbox, and during my time at Georgia Tech, I have previously worked on multiple projects, some of which are highlighted here, both trying to develop new methods to study microbes, such as optimizing metatranscriptomic protocols or reconstructing whole genomes from sequence data, or studying viral sequences to understand the role of viruses in the environment. But also I have applied those techniques to understand the role of microbes in important processes such as nitrogen cycle in the ocean or biodegradation of toxic chemicals. And I switched gears again when I moved to Emory almost 3 years ago to join Dr. Brunner's team and apply those ecogenomic methods to study microbes in clinical settings and understand how they affect human health with a focus on cancer populations. Some of the projects we're working on with a lot of different collaborators include looking at the microbes in the human breast and see how and if they play any role in cancer or treatment outcomes. We also studied the gut microbiome of mice that undergo immunotherapy, trying to figure out how the microbiome can affect treatment outcomes as well. But the main project I'm working to, and I will give a very brief overview in the next couple of slides, is the vaginal microbiome in postmenopausal women that undergo cancer therapies for gynecologic cancers. And we do care about postmenopausal women. We know that they are affected by what we call the genitourinary syndrome of menopause, a result of low estrogen levels and vaginal wall thinning that results in symptoms such as dryness and dyspareunia. On top of that, gynecologic cancer survivors, which are almost always postmenopausal, are experiencing additional symptoms caused by the cancer treatments itself and especially radiotherapy, as Zahra also briefly mentioned in her introduction. We also know that the vaginal microbiome plays a critical role in the vaginal health. However, associations between the vaginal microbiome and cancer treatment toxicities have not yet been established. And actually, at this stage, we really don't even know what a healthy postmenopausal vaginal microbiome should look like, since this population is largely understudied. So our overarching aims for this project is first to assess if there are differences between healthy people and people with gynecologic cancer, diagnosed with gynecologic cancer, and that also taps into the question posed before. Is it the cancer or the cancer treatment that has an effect? But also we want to understand if there are any associations with the symptom that caused by the cancer treatments. And for this, we recruit patients in our gemstone study, and both Gia and Zahra have worked on this. And our study includes two arms, the gynecologic cancer cohort, where participants are monitored for 1 year before their treatment, after the treatment, 6 and 12 months later. And also we have a healthy cohort of participants. We are trying to match them in age, BMI, and ethnicity. And we will monitor at the same time for understanding maturation effects and changes in the vaginal microbiome that are not related to cancer and treatment. On one hand, we collect vaginal swabs and we perform all the bioinformatic analysis on the microbiome data. And on the other hand, we keep track of clinical factors and patient-reported questionnaires where we're trying to capture all the vaginal symptoms that people experience at different times. The goal is to identify associations between the microbes and symptoms while controlling for various sociodemographic factors. Finally, I will just go briefly some key findings that this analysis have yielded. In this preliminary data, we had about 100 healthy and 76 cancer patients with the different time points. First, indeed, we saw that symptoms are more prominent in the cancer cohort and as expected. You can see with the red is the cancer people and the green is the healthy. Cancer patients not only report more symptoms, but also persistence of symptoms. Some of the, for example, people that report persistence of the same symptom for a year, it's about 23% of our cancer population, while it's only 12 in the healthy cohort. Second, we have seen differences in the vaginal microbiome as well. And the key findings here are presented on those two publications, one under review. So the major conclusion from this study is that postmenopausal women have a large variability in their vaginal microbiomes, and lactobacillus dominant communities are rather the exception rather the rule, as opposed to young women. What you see here in this dendrogram, each bar represents one subject, and the taxonomic distribution of the vaginal microbiome is shown. For example, some people have vaginal microbiomes that consist almost entirely of lactobacillus, which is here in pink, but the majority do not. They have multiple low abundant species, and when we cluster those microbiomes based on similarities, we can identify at least 7 different types, for which we call them community state types. And some of those types we actually did find to have strong associations with symptoms such as lack of dryness, specifically for the type E, F, and G, or abnormally high pH. Finally, we also look at individual taxa, and we see that cancer vaginal microbiomes have about a dozen phylotypes enriched, and those are typically associated with various pathogenic states. And moreover, we also detect the effect of the treatment itself when we compare our cancer population before and after treatment, and we see that after the treatment, we have several low abundant species pick up in our cancer population. This is typically a sign indicating a perturbation in the microbiome. We also see that through time, and we also observe striking difference between cancer and healthy in terms of their alpha diversity. And finally, we also see that another significant finding is that when we look at the 4 different time points, cancer patients have extremely unstable microbiomes compared to healthy people. And this is what you see here. It's a metric of stability. The higher it is, the more unstable or the more changes the microbiome has through time. So you see that the cancer patients have the more unstable microbiome compared to healthy controls. And overall, those two are postmenopausal women, and those are reproductive age women from the literature compared at different time points. You see that postmenopausal women, to begin with, they have higher instability in the microbiome, which is typically a sign of perturbations. And finally, despite the small sample size that we're dealing here, we have been able to detect associations with symptoms as well. For example, we saw specific species such as Delphtia correlate with vaginal pain, and the lack of lactobacillus or high abundance of Prevotella and Fusobacterium correlate with persistent dryness on those women. To sum up, we have seen evidence that both the gynecologic cancer and the treatment have detectable effects on the vaginal microbiome, and that cancer patients have characteristically unstable microbiomes with some opportunistic pathogens. And this instability tends to associate with symptoms such as dryness and high pH. And moving on, while those results are promising, they are based on a small sample size and on 16S analysis, which, while valuable, cannot identify species within the microbiome, but rather higher taxonomic ranks. So our next goal is work with metagenomic sequencing in order to not only identify species at the higher resolution, but also the functional potential of those microbes, and begin to understand how they impact symptoms and treatment outcomes. Some of our current work right now that is in progress includes method optimization, such as optimizing a protocol to quantify bacterial load in the vaginal sample, which is which might be an important factor, and this is something we cannot get with the 16S. Testing methods for obtaining high quality metagenomic data, which is a huge challenge in vaginal swabs because we have a big contamination of human cell DNA, so it's really hard to capture the microbes. And finally, we just starting a study monitoring shorter dynamics of the vaginal microbiome in postmenopausal women in order to understand the factors that affect the microbiome on a daily or weekly basis. And to close up these presentations, I put a generic study scheme here of the gemstone study. This is actually, we just submitted an R01 on this, and we have submitted several smaller and foundation grants on the various aspects of this project. But the big picture is that at this point, we're studying just associations with symptoms from the treatment or from the cancer itself. The next step is to first expand our study in associations with cancer recurrence and HPV infection, for which we have some preliminary data. We work with our CDC collaborators on this, as well as local inflammation by looking at local inflammation markers in the vagina. And outcomes from those associations, for example, which specific microbes are correlated with specific outcomes, will be tested in vitro settings in the lab to reveal the mechanisms of action, such as cell cultures inoculated together with specific bacterial tags. And of course, understanding the functions of those microbes in the context of cancer treatment and patient experience can eventually lead to therapeutic and diagnostic tools to improve patients' health, which is the ultimate goal. And thank you very much for your attention.
Great, thank you, Despina. Wonderful, wonderful work. Really great to see the results coming to fruition here after your time. What questions for Despina?
Hi, Jana.
Hey, Sunwa.
Yeah, I have a question for Despina. Despina, thank you for this beautiful work that you have done since your PhD and then continuously at your postdoc by adding more, cancer relevant results. I think your framework is very exciting. I think that's only your first few slides. One part I think is interesting is for the single cell. So I am, I feel like single cell, sequencing is very exciting. And so how are you connecting this single cell with, microbiome data?
Yeah, that's a good question. Thank you, Sakamba. We haven't applied that technique yet in our samples, but single cell sequencing is very valuable when we cannot isolate specific microbes, either through bioinformatic approaches, such as binning their genome from sequence data, or trying to isolate them in the lab. So what we can do, we can use this technique in a sample to sort cells one by one, isolate them, and sequence them independently. And I have previously applied that technique. It actually resulted in a Nature publication where we were trying to isolate microbes from a specific Olsen regions, oxygen minimum zones, and we couldn't. So we used that technique to isolate them and study their individual genome separately. But yeah, this can be applied virtually to any sample as long as you store it properly. And it can be very powerful when all the other ecogenomic approaches cannot give you what you want. It's when you're looking after a specific microbe.
Okay, so you are using the single cell scale not for the cells in for a human. You are using the single cell scales for the bacterial cells. Is that what you?
It's for the bacterial cells, absolutely.
Oh,
yes.
Okay. Yeah, thank you for this. And because I was thinking of using some like a single cell scales in now stored PBMC, human blood samples. And so, okay. So maybe I feel like Yufeng maybe in the
I'm not
future.
sure if you have enough bacteria in the blood sample.
Oh no, I'm not talking about for the bacterial. I'm just talking about for the human single cell sequencing of.
Oh yeah, this is a completely different.
Different, yeah. So I was wondering how you are using that for the bacterial. That's very exciting. Thank you.
Thank you. Any other questions? Okay, so I think there's a theme here. Although our postdocs are all working in some pretty diverse and different areas of science, the postdoctoral year is really meant to grow your research independence, to publish, to start getting competitive funding, to network, to find people who can help help shepherd you through your independent independence trajectory, and start to make a name for yourself, and start to do something unique and innovative. And I'm regularly impressed with you guys, but after today, even more so with the great work that you've been doing. And I wish you all the best. Thank you so much for your time and your energy and your contributions to our school and your contributions to science and, my gosh, the well-being of the world. You guys are wonderful. Thank you all very, very much.